All right. Hey, everybody. Good morning, good afternoon, good evening. We're going to wait for a little bit for folks to trickle in. I think people join from all over the map. Like we've seen East Coast, West Coast, some Europe. Welcome to today's Tech Talk. All right. I am delighted to bring you the first edition of our webinar series in 2025. My name is Devika Garg. I'm the Director of Product Marketing at TileDB. And I'm joining you from sunny and also kind of rainy California today. I'm joined by my colleague Meher Hamdi, who's ensuring everything in here goes absolutely like per plan. Thanks, Meher. And then today, like we have the privilege of hearing from Sam Cooper. He's co-founder and chief technology officer of Phenomic AI, and he'll be sharing a remarkable journey in revolutionizing cancer therapy research with advanced single cell work. Sam, it's so great to have you here.
Oh, fantastic. Thanks, Devika. Yeah, good to be here.
Where are you joining us from?
I'm joining us from the Netherlands right now. Yep.
Oh, nice. Okay, so it must be, yeah, it's afternoon almost or near evening for you.
Later.
Yeah. Well, I had the pleasure of meeting your PhD advisor, Chris Pakal, in London just last week, and he was speaking so highly of your work in computational biology. So thank you again for being here.
Thanks very much for having us. And yeah, I look forward to talking about what we're up to.
Absolutely. Yeah, I mean, so this is going to be very exciting. We're going to talk about how Phenomic AI uses ML and TileDB for scalable single cell analysis. And I believe like, if you're joining us from a biotech, early stage, mid, or pharma background in data science role, technical leadership position, I think you're going to find this valuable. A couple of housekeeping notes. Please add questions in the Q&A panel at any time during our talk, and we'll tackle those at the end. And we also have a raffle towards the end. So if you stick on all the way, like there's a chance you might win a $100 Amazon gift card. Always nice. All right, let's get going. We have a quick opening poll that we wanted to do. Sam and I both wanted to do. In a little bit around, are you actively developing therapeutics? Are you a biotech pharma? And what is your level of experience with ML techniques in single cell? Let's see what we get. Maybe we allow like 30 seconds, 40 seconds. How is it looking, Mayor?
We should be nearly done here.
All right. There we go. So, okay. So it looks like we have a lot of folks who may be like tech bio folks, right? Or what do you think? Like the nose, Sam? Any comments here?
Yeah, I don't know. Yeah, probably a lot of people doing sort of software development and software applications there. So yeah, good audience. Right. Yeah, would be good to see. We'll get into some biology, but I'll keep it pretty technical, I think. Then yeah.
Okay, awesome. All right, thanks. Let's close this and keep going. All right, so agenda for today is, we're going to talk about, just excitement of single cell biology. Why is this so important today? And touch on Phenomic AI's approach. And lastly, talk about, data foundations, what kind of things are important in when you're leveraging single cell data for drug discovery. So Sam, I'll kick this over to you. Like, what is so exciting about single cell biology? So,
well,
yeah,
thanks, speaker. Yeah, I guess happy to jump in. So I think what got us excited sort of Phenomic for single cell biology is we think it's sort of really the first sort of technology that's allowed us to understand what sort of cells look like and sort of what's that full set of cell states that exist inside sort of solid human tissues. Fantastic being able to look at sort of flow data for understanding sort of blood cell types, and lots of histology data has been great, but none of this is sort of that whole genome resolution that you really want at the single cell level to understand sort of what state of cells existing in. Sort of single cell has definitely come along, totally changed the field here. My background was actually in sort of analyzing sort of imaging data. Single cell data, by comparison, just the sheer amount of data you get out, especially understanding sort of cell state, is just vast, orders of magnitude more than you get from the imaging side of stuff. So it's been super exciting to sort of build out, sort of shift phenomic around and build out in this single cell world. I think we're sort of scratching the surface of what we can achieve here. Really sort of on the tissue side, definitely getting huge amounts of data now, definitely sort of hitting sort of peak, sort of output here, I think, and peak understanding from, especially human biology. As we're getting into sort of, what's happening now, and I think sort of the next sort of 5 or 10 years is all going to be sort of single cell applied to in vitro experimentation, increasingly complex models. So, in vivo biology, human tissue explants, there's groups now out there even doing sort of single cell analysis of like your entire live organs, that have been sort of discarded from sort of transplant surgeries with sort of drugs applied to them. So definitely scratching the surface in what we can use for functional readouts, but in peak sort of period for understanding sort of definitely human tissue biology.
And I mean, single cell biology also being used in screening right now, like is that, how prevalent are you seeing that? Is it just starting, or would you say?
Very much the start of the wave there. Yeah. So this is what we're excited for. I think, definitely is, we'll see where the data really sort of cranks in now, is that, traditional sort of screening outputs have always been sort of image-based, or, functional basic sort of functional assays, or, just sort of single ELISA stuff like this, single dimensional readouts. Image analysis, sort of high content imaging, this really pushes, pushes up the dimensionality you can get out of one of these screens. But challenges, especially in high content data, abound about how you can relate it down to sort of fundamental mechanism, how you can map that to, is that sort of an effect we actually care about based on human tissue biology. What's super exciting from single cell data is, we can definitely directly map sort of, what we're seeing in the dish to, what we're sort of hoping to see, based on what we expect, a good outcome in the clinic would be. So that ability to relate sort of in a dish biology through to human tissue biology is, incredibly powerful, and something you just can't do with any other approach. Plus just the vastly more data you get out of a single cell sort of screen, really means this, I think, will be the future of, well, the next sort of 5 or 10 years of screening are going to, see a huge surge in sort of single cell approaches.
And I mean, it kind of ultimately sounds like it is reducing failure rates, which is like the early failures in reducing that a lot.
Yeah, well, our hope is that, by getting all this sort of mappable single cell RNA data out, that, we can, I wouldn't say, increase the failure rate early on, or just, figure out what are those most relevant targets much earlier, versus having to, wait till sort of later stage models, or wait until, clinical readouts to get that sort of, mapping to real relevance. Yep.
Yeah, I mean, let's dig into your specific, what you guys have been doing at Phenomic, which is, working on solid tumor biology, which I believe is really, really hard. And so, yeah, let's talk through that.
Yeah, thanks, Vika. Yeah, so just, for everyone watching as well, next couple of slides, just going to dig into a bit what we're up to the company, but also provide some sort of, hopefully technical and interesting sort of take homes on how we're thinking about single cell data. Just briefly as a company, so our sort of focus is going after sort of solid cancers. We're a full biotech company. We're developing new therapeutics ourselves and have our own sort of pipeline, as well as some really awesome partnerships with sort of other companies looking to do sort of looking at similar biology. In terms of our sort of thinking, what we sort of think is that, and a lot of this has come from single cell analysis, is that sort of solid tumors are way more on a spectrum with sort of fibrotic diseases than, is often traditionally thought of, with them being, coming from sort of more mutational, people coming from a sort of more DNA sort of mutational background. Where you look at like hematological or blood cancers, these are definitely on the far end of like the mutational spectrum. One mutation can definitely probably trigger a hematological cancer, especially in the right environment. You just have to get very unfortunate there, and it happens. Solid cancers, you see it way more associated with sort of late stage, with aging. You see it associated with fibrosis. If you've got chronic fibrosis of the liver, you're way more likely to develop liver cancer, organs of magnitude higher. We think that, what we're seeing is that, a lot of these solid cancers aren't necessarily straight from a single mutation. It's rather that, that local microenvironment due to, fibrotic disease has become like, ripe for cancer to take off. then that cancer feedbacks and drives further fibrosis. And it's really this sort of feedback loop between fibrosis and, epithelial cell growth that's driving your progression in these diseases and makes them so hard to treat. So what we're focused on in Phenomic is really trying to understand this. Ourselves, especially in partnership with others, are looking at how we can break this loop between sort of fibrosis and cancer. Our internal pipeline is focused on sort of how we can take advantage of the fact there's tons of fibrotic tissue around cancer and use this to selectively target sort of payloads to kill the cancer cells themselves. But this is our sort of thinking, and this has all been informed by sort of looking at lots of single cell data.
Okay. And so I'm controlling the slide, so please feel free to let me know when I.
Oh, this is great. Yeah, thanks. Yeah. So in terms of our pipeline, sort of how we go after the targets. So we built up this massive atlas of sort of tissue data, based on single cell RNA. This is what we're hosting in TileDB. What we're doing based off this is identifying sort of assays and models that we think really reproduce sort of the tissue biology we're going after in humans. We're running sort of tests of our own drugs, running screens in these assays, figuring out what works, seeing what we think is going to have the biggest impact on specific patient groups, or really as many patients that we can go after. And then we're now taking stuff that's sort of working in here, developing drugs for it, then going directly into sort of human tissue explants. So this is sort of live chunks of human tissue that we're getting from the sort of network of hospitals that are participating in a program we're part of. And we do own test our drugs in these human tissue chunks. And try and look for signals of efficacy that we expect to get out of, that we're expecting to get out, of patients in the clinic. So this is what's sort of informing our pipeline. We try and keep it as close to the human tissue biology as possible, and it's all informed by sort of analysis of single cell data.
Right.
Awesome.
And it's a lot of single cell data, right?
It's a lot, yeah. So we know there's sort of other curation groups out there. We were one of the first to sort of just set off and just start curating huge amounts of single cell data. We've now been sat on this for like a number of years. We've thousands of patient samples worth of data from single cell biology. We've brought in additional sort of spatial stuff too. We've got a bunch of samples from murine systems as well, and increasingly now a lot of our own data as well from sort of screens and sort of these explant systems we're running. We're very focused on cancer, so we basically got, as far as we know, basically every single cancer data set out there, kind of standardized with the metadata, aligned it, made it all nice to sort of manage and understand. And some summaries here, we see a nice spread across all the sort of key cancer types. Overall, in terms of like just number of cells, I think we're in like tens, if not coming up to 100 sort of million cells now, is my understanding.
Wow.
I think it's way up 100 actually. That might be a bit of a lie. I think it's more like 20, 30, 40 sort of zone. But yeah, we've got a lot.
That's a lot. And talk us through the data journey in general, like how much of this is public, and then you mentioned a lot of partnerships with hospitals and providers, and so you're getting those samples, and are you like doing all of those experiments in-house or working with third parties, and like if you could like shed a little bit of light on that.
Yes, we're generating all of the like sort of tissue data in house. We've got a couple of scientists down in our labs in Boston who are working tirelessly at whatever out of date the tissue arrives to help us sort of profile this. So absolute hats off to them for their hard work here. It's definitely a bit of a barrier to entry for many. It takes time. It takes lots of filling out the right protocols to make sure that we're doing good work and work that patients would value. So we ticked all those boxes. We're happy to be in this place, but definitely anyone else can work on this as well, and think having access to these sort of if you can get access to these patient samples, it really creates a definite advantage in being able to figure out the best biology and drugs to be working on.
And there's no cell lines here, right? It's not nothing cell line based.
Cell lines is always like a bit of like everyone's like black and white. Oh, it's a patient cell, or oh, it's a cell line. It's such a spectrum. You've got sort of cells which you just get straight from like the most sort of primary ones out there, ones straight from your patient. They're often there. You hit tissues as like donor variability and stuff, but they do if you get the right sort of cell type, they often look most similar to what we see in patients. On the fibroblast side, that is certain immune cells as well. Cancer side, cancer cells just are naturally immortalized. So is it a line? Is it just it's pretty similar to what you're getting straight out of a patient. Probably a lot of these lines. It varies depending on what question we're asking, what we're doing. We try and keep it as what we do is we just profile stuff by single cell data and then try and say or sometimes bulk and say is it expressing the genes we think are causing a problem based on human tissue data? Is it matching that phenotype that we think is disease driving in patients? And if it is, then it's a good experimental model for us.
Great. That makes sense. All right. Should I keep moving?
Yeah, definitely. Jump on. So there's some technical stuff. So if anyone's in the world of single cell analysis, the next sort of slides are for you guys. So one of the sort of early challenges we sort of faced, and this is what we spent a lot of time trying to figure out, is how we can sort of align lots of data from different studies. So instead of having to go through every single study one at a time, when we run big analysis, we want to put them all into a single database. TileDB solved this part. We wanted also to be able to make sure that it's the same cells in one study to the next study that are being called the same thing, and manifest other things like that. We can assign the right metadata across studies. Do the right sort of queries. So we wanted everything very nicely aligned into the same sort of space, so we know what sells what. And that there's no sort of technical batch effects driving certain differences between cell types and stuff, or between studies. So if you look in the world out there, there's a couple of sort of good sort of comparison publications, especially from sort of Fabian Dice group. These tend to focus more on sort of picking what model is best for a certain problem versus strictly what model is the best model out there for aligning data at a massive scale. We ended up figuring out our own sort of metrics of there weren't even good metrics to say how well can you align, like a good single metric to say how well can you align massive amounts of data. So this graph on the left, we created our sort of own score for this. It's a derivative of this score called the K-bet score. Again, came from Fabian Dice group. We sort of tagged this up to a score for accuracy to be like can we accurately get cell type label correct? And you have to hit both these tasks. So we're looking to try and simultaneously get rid of batch effects and make sure that we get cell type label correct. So this sort of graph on the left kind of highlights the sort of different cases. So when we're looking to align data, we want to basically make sure the cell types overlap with each other and batch effects are gone between different studies. The score we created, so we created this sort of benchmark data set. We published this data set. You can go, it's a preprint at the moment. You can just look at my name or have lead author at the moment. Lead author is Javier, one of the team members in our company. You can go Google our names. You'll see the paper. It's on bioRxiv at the moment. We released the data sets too. So we created this massive benchmark data set of 48 different studies. With some smaller sort of benchmark data sets as well. But the key thing we did is we kind of just manually made sure that all the author names overlap with each other. So the idea is that you can use all the other data sets to predict what a cell type label is in a held out data set. So you can say, okay, if I've aligned all these data sets well, I can predict one of these cell types from the missing data set, get it right. And then then I've aligned the cell types with each other. I can also say, does that look like all the other cell types, or does it look like it's only cell types from that study, in which case it's a batch effect. This score on the left sort of measures it. And if you look like you're mixed up with other cells from other studies, and you are in the sort of right neighborhood with the right cell types, then you get a point. So this is the score in the middle here when we tested a bunch of different models out there about how well they align different single cell studies. Some of the more basic ones are like Surat is RPCA. You've got sort of basic skivvy out the box here with 2 layers, skivvy 2L here in the middle. We basically figured out how to really optimize this alignment procedure. And we used this sort of adversarial trained skivvy model there, BA skivvy, BW1K in the middle. This is what we found is sort of the top performing model with some tweaks that we made for aligning single cell data at massive scales. And the effect of that there's on the right. One thing to note, these newer foundational models we found basically they sort of bake batch effects in to the alignment. So they cause batch effects and they create problems. So we've stayed so far away from anything like Geneform or SCGBT. This might change if they can improve, get rid of the batch effects at train time. We've not seen that yet.
I see.
For example, Geneform here on the right, you get this batch effect still here. This is looking at the study label here. And there's this sort of NKT cell population here. We see this group as EZAL is sort of this one particular study there. it's this really dark blue area. This is a clear batch effect. If you take the sort of more, aggressive approach, well, this sort of more adversarially trained approach that we do here, BA-Skivvy, you can totally get rid of that batch effect. And if you look on the cell type space, I've not got it here, you then see perfect alignment of NKT cells as well, not driven by batch effects.
I think that is really important because with SCGPT and Gene Farmer are out there and I think used quite a bit. And so would you say that is there could you actually have a foundational model for single cell data like this, or does everybody need to train their own? And is there
so foundational models is kind of like the current language for a massive transformer model, and they're not always best. What we found empirically so far is that you can embed you can get a lot of information in quite a small space. So maybe the amount of information, the number of cell types doesn't yet maybe require a massive transformer model. There's definitely ways of arguing this, and I'm sure as you bring in extra modalities, it sort of changes. There might be better, there's probably better architectures and especially sort of language-based transformers for managing single cell data at scale. I'd love to see some smart approaches using, you could probably do some smart approaches with a transformer architecture, but not in the way we've seen them so far.
Yeah. So folks could take SCVI in their own environments, test it out.
Yeah, the best we've seen, we've published the approach to we train this BA-Skivvy model here. I think there's even an adversarial function now in the core single cell variational inference package. If you just turn up the number of layers in your single cell variational inference architecture up to 3 or 4, you usually get quite a lot nicer results, just out of the box as well.
Very cool. Very cool.
Awesome. And so just highlighting, so this is the full benchmark now. So this is from 48 different studies. And we compared, this is our score here, the KNI score on the left. Again, you see that, really the top mark comes from this BA-Skivvy architecture. In this, we've done actually, some of the labels have been mixed up here. This is a GF BA-Skivvy is the one in pink. Yeah, I've got some mixed up labels here. But you can see the UMAPs here on the right, and we've got the real numbers in our paper on the internet. But you see again that so this is SCGPT slightly overfits here a bit actually. We found the Gene Farmer has a ton of batch effects in it. We're still getting the best results of this adversarial trained single cell variational inference by quite a margin. What's kind of cool as well is you can kind of reproduce the groupings in it. So this is 48 different studies in this big benchmark here. You can take any one of those studies, you can rerun UMAP on just that study, and you basically get the same original cell type groupings and labelings out that the authors had in the original study. So we don't think we're overfitting here as well. We're just really aligning everything into one fell swoop. So take home is Skivvy's great, especially go up to sort of 4 layers or introduce adversarial training.
Makes sense.
Cool. Sweet. So jumping into different gears here a little bit. So what can you do with all this data? So this is sort of the biology side of things now. A bit sort of higher level and going through a bit quicker here. So as a company, we're focused on this protein CTHRC1. We're super excited by it. Based on a huge analysis of fibroblasts in cancer and normal tissue, we basically see it, in single cell level as the most upregulated target in all of cancer. It really stands out. And this is this x-axis here. So this is analyzing about 500,000 fibroblasts across about 2,000 sort of patient and normal samples. For solid tumors, CTHRC1, the most upregulated gene. We can do these sort of in vitro co-cultures as well. So we can take patient calves, stick them alongside cancer cells. We can do that with other cell types. And we find that CTHRC1 really is uniquely upregulated by these sort of cancer calf interactions, direct contact between the cell types. This is an atlas visualization on the right here as well. So this is then breaking down our atlas by tissue type. Each dot on this point is actually a patient sample. So there's not single cell histogram. This is that pseudo bulk sample for fibroblasts specifically. So you're looking at the average sort of fibroblast recount per sample. And again, you can see this huge upregulation of CTHRC1 in cancer. We looked at sort of fibrotic tissue as well. Not quite, nothing on the same order of magnitude as you get in cancer. Although most target, it's a really fascinating target. It looks very selective to cancer. You see it a bit in sort of very early in sort of embryonic tissue. It sort of is involved in patterning some epithelial cells before they start setting off on gastrulation movements. But yeah, other than that, basically very clean. So super excited for this target. And it's sort of
Is this a net new, like novel target, Sam?
yeah, there was like some other groups. A couple of people have sort of mentioned before. It's been called out in fibrosis, but I think we're the first to figure out how selective it is and how like it's just up in every single solid cancer. And this is unique to the amount of single cell data we've aggregated.
Very awesome. Yeah.
Yeah, no, another cool good take home. If you guys are looking at the human protein atlas single cell data, that is like take it with a very big pinch of salt. We've got that's like very much based on individual studies, and some of those studies are a bit wonky, and some of the alignments are a bit wonky. You're way better to try and either take an approach like we've done and really aggregate single cell data, or look across a lot of studies on like cell by gene to get an understanding of sort of single cell expression. Yeah, but yeah. So this is, we then validated this target. So we've identified sort of, all the antibodies out there are pretty terrible, but we developed some very selective antibodies for immunofluorescence. We validated these with like knockouts and cell pellets and stuff. Very selective, and we show very, very nice localized staining of CTHRC1. And what was actually fascinating as well, we didn't get this from the single cell data, we had to go figure this out. We found out that CTHRC1 is being produced by cancer-associated fibroblasts. So it's secreted by these sort of stromal cells, but it actually sticks to cancer cells and sort of coats cancer cell, the membrane of cancer cells. So this is when we got really excited because we're like, oh, we could use this to target, T cells to go and attack anything with CTHRC1 stuck to them and get great killing.
Immunotherapy.
So what we've done after a lot of complex engineering, really awesome work from the team. On the next slide, I think it will show the mechanism. Yeah, so we've got this really cool T cell engager now that basically this sort of CTHRC, one of these little like blue sort of club-like things, is being produced by CAFs. It goes and sticks to the membrane of cancer cells, and then we can cause T cell killing of cancer cells themselves via our drug, which is bridging CTHRC1. And so the effect of this on the right here, this is a lovely little in vitro assay using sort of some patient CAFs and PDAC, co-cultured with KP4 cancer cells. And the effect is that, we can induce killing of cancer cells. Basically, you get CAF-dependent killing of cancer cells with a T cell engager. So if you have got no T cells, you don't get killing. If you've got no CAFs, you don't get killing. You have to have both there, but then you get very potent killing of cancer cells, which is very, very exciting. We've now shown this in vivo. We've shown this in tumor explants in a dish, and we've done some sort of initial safety studies. So we're also looking to fundraise for a phase 1 trial. So if anyone wants to give us some money to go run a trial in humans, you're on the hunt for that at the moment. Yeah, but yeah, very excited, for this drug, and should be in the clinic in sort of 2026.
That is so exciting. Like, and I think the really cool part is like this would work in all solid tumors. So that
yeah, if it works, it's gonna knock it out of the park. I mean, yeah, if it does work, it'd be an absolute blockbuster. It's it's a new mechanism. No one's done anything like this before. But if it works, it's gonna be absolutely spectacular.
Amazing. Fingers crossed for you guys.
And then the final thing, so I think the other thing you can do with single cell and some of this sort of RNA data that we're really excited for is being able to define translational strategy. So going beyond just single cell RNA data, we think that you can get pretty seamless sort of embedding or seamless sort of integration of all the RNA data modalities. Certainly, you can imagine that like this sort of spatial RNA, each pixel is just a collection of single cells. So you can sort of pseudo bulk up single cell data to create, to draw that line between single cell data and spatial. You can then pseudo bulk across the whole sample level, and that's how you get to bulk RNA. And that really we think allows us to integrate, create this sort of alignment of all these sort of data types to identify specific patient populations. We can go and test in sort of in vitro in the right models and our explants, drugs in across a bunch of different patients or donors, and then we can say, okay, in this group of donors or this group of patients, we see the best efficacy. What indication does that map to? What biomarkers does that map to? And this is how we're thinking about picking specific patient indications or populations and specific sort of biomarkers to understand potential signals for efficacy in the clinic. So yeah.
This is so amazing. Like my head is spinning. There is so much different data, so many different modalities, and the scale is tremendous. The variability is so huge. I mean, I think we have to move to our next section on foundations. Like, how do you get there? Like, this is amazing biology, right? How did you get there? Like, sure, it's not an afterthought, right? And so what kind of things did you think about early on, Sam, at Phenomic, like when you were just starting this journey? Let's talk about that and then we'll kind of dive into a little bit more just specific to single cell.
Yeah, definitely. Sounds good. Jumping now. Yeah, so I guess I can jump in. I don't know if I can't even remember what slide's next, but this one's fine. Yeah, in terms of how we first thought about it, yeah, so we just started going. Like we didn't have like a grand plan. Our plan was basically like if the more single cell data that we get, the better the targets we're going to pick. So there's a Javier on our team and a handful of other, sort of others, especially some sort of part-time people as well. You did an incredible job of starting to create huge amounts of single cell data very early on, before it's cool and before other people were doing it. So this data very early on helped us figure out what to start going after. We started hitting scaling blocks along the way. This is where, some awesome coders on our team, Dylan, Octavian, just off the top of my head, others, as well, really helped us, figure out how to deal with the scaling. Along the way, we were like, how do we store the data? And this, where we came to you guys being like, okay, this, empirically seemed to be the best solution, based on some tests for storing our single cell data and without huge amounts of coding on our side as well.
Yeah. And like we see like on our end, a lot of different kind of challenges with single cell data, like either at scale or even at lower levels, that there's all these new assays that and evolving, they're continually creating data sets that are too large, or there's too much variability across different methodologies or different algorithms that are available for analysis, or and there's a lot of custom formats happening. And so like when you're trying to, it's one small experiment, every single experiment becomes a silo, and combining all of that together, right? So it's all this lack of interoperability that hinders all of that. And I don't know, like we've also seen that file-based approaches to this kind of data also like creates the same kind of problems with there's no fair data, that framework is kind of almost missing, and groups cannot collaborate within companies. I don't know if you guys saw this early on, like as you started to hit that scale.
Yeah, so for I've definitely seen this in other groups as well. For us, we sort of try to maintain like rough standardization organization. I mean, this is what we're solving. All the data out there that's published is in its own paper and its own sort of repository. We're starting to see. I mean, I think you guys, Cell by Gene's based on you guys as well. So they're one of the good public efforts as well to sort of start to solve this. The challenge you get, definitely is that like, you can keep stuff in like, I think top point there. The problems we face, as you start to get bigger, memories, you can't keep it in memory. It starts bombing out the second you want to do anything in memory. If you want to try and like manipulate or move the data around the place, it becomes increasingly unwieldy. So we were just excited to have like a solution that allowed it to that lets us like put it all in one place in one format and be able to do big analysis without things breaking down unnecessarily.
Great. And I mean, I don't want to go into this for too long, but just a little bit about, why TileDB is has been really key for single cell data. It's a database based on multidimensional arrays, and it gives you the flexibility to tackle a lot of different modalities. And just in the life sciences, for example, we have applications for imaging, and so you can store pixel data in densities indexed by XY and the channel, depending on the format. And then for VCF, you can store each variant along with its attributes in a 3D sparse array. And this is indexed by chromosome position, sample ID. And then for omics data like single cell, you're working with large and very sparse 2D arrays where each value is indexed and by its feature and sample ID. So like with this, just one, this kind of multidimensional array model, you're able to condense into different formats that you're able to address different kinds of data types and applications. And specifically for single cell, like it solves all of those problems. It's scalable for Atlas scale level, and interoperable between different analysis languages and packages such as Surat, Bioconductor, ScanPy, and it is optimized for object scores. So you don't have to download this data locally and you can work with it. And lastly, like it is multimodal. So again, like the different kinds of data that you're working with, you can all bring it all together into one space. And then Sam, I think you have picture of, how everything comes together, like the curtain, lift the curtain, behind all of the wonderful data type.
Yes, we've got sort of two slides, I think, here on just showing how we do it at Phenomic. So this first one, sort of the higher level slide in terms of just like, where sort of all our data lies. So we keep all of our single cell data in TileDB. The sort of machine learning stuff is sort of more run directly against the sort of TileDB database. It doesn't really see sort of the front end on the biologists. All of our machine learning work is really trying to understand how we can better align the massive amounts of data we've got. Now we're looking across species, not just humans. Also looking at how we can potentially infer, different types of metadata from sort of what does exist versus what doesn't, i.e. is a tumor sample T-cell infiltrated or not? Do we think it's going to respond to immune therapy or not? So this we do sort of all on the machine learning side on the back end, and that feeds back into this sort of TileDB object. This sort of serves then, both biologists and informaticians working in the company. The software guys on our team have done a really great job of, putting this all under this sort of Kubernetes sort of system that allows you to sort of scale based on demand. So we're not paying huge amounts of cash always just to host giant servers. On the biologist side, Octavian and Dylan and Elias coded up this great, user interface. Basically, sort of got React in there, couple of other sort of libraries we use. And this allows us to visualize sort of all this massive data set of single cell data basically on the, in real time, you get sort of quite, single, sub 1 second sort of queries of, tens of millions of cells across all the indications and samples. This allows you to say, oh, is this target up in these specific indications? Is it down in these indications? What cell type is expressing it? And you can also run differential gene expression. I think we've even got like, we've got, it's, I think scref.phenomic.ai. You can have a little look. We've got a little variant of this actually just on the internet that anyone can use for normal tissue data. That's mentioned in our bioarchive paper as well, I think. So feel free to go play around with it as well if you're interested in gene expression. But really powerful interface for biologists. And we're increasingly now shifting into this world where when they what we want to get to is where they can run an experiment and then almost instantly it goes onto the user interface and they explore the results of their experiment. On the informatics side, this is sort of what the bioinformaticians in the company mainly work with. We've got sort of notebook environments that allow you to run sort of powerful analysis of the TileDB data set. We've got our own sort of optimized libraries for DGE across tons and tons of massive amounts of data. And then we've also got APIs that allow you to take sort of new wet lab data, send it off to our server, get back embedded data based on machine learning models that have been trained on the big single cell atlas as well. So we've now got to this pretty cool situation where we've basically got real time, on the fly, embedding and annotation of data very accurately, and getting towards the point where biologists will even just get the report straight back that they can sort of act on.
Very nice. And then
This is in the weeds ones. This from one of our coders, Dylan, did this lovely chart here, and this is the stuff he's been working on. And this is how we actually get raw data into the TileDB Summer object. So we basically store everything in like the rawest format. We have sort of just straight MTX files or AData objects. This is like, these are great if you wanted a tiny number of, if you're happy handling just like 1 or 2 experiments of tissue types. As you really start scaling, they sort of start breaking, and then you have to, you just can't really analyze them anymore. So what we've now got then is Dylan's built up this sort of script and the system that allows you then to just on a quick trigger, and this trigger can be anything, can be automated, allows you to put all of this data, basically fires off this whole system on the right here. Then we're using sort of concurrent, the concurrent write sort of capability of TileDB to quickly build up this big Soma object that then goes back into all this app and all these applications and stuff that we're using for analyzing the data.
This is really nice. I'm hoping a lot of folks will find it very insightful. So, I mean, I think you've probably covered this, but if, this is my question that if not TileDB, what else? And what was the selection factor here for like picking TileDB for all of this data for you guys?
Yeah, so I think, we were sort of weighing it up versus, coding up our sort of own SQL database where we'd have like, a row. You can like, for example, store sparse vectors as, recount to sparse vectors, and then just store them in the SQL database and do a, analysis of that. It would require significant coding from our side, and a bunch of effort, plus probably be not as good. So we just tested some of your guys' approach and a few others to see what handled our sort of single cell atlas the best. And we were like, okay, this is great. It was easy to work with, didn't need any coding our side really, and gave good results in terms of performance. So we were happy.
Please let me hear that. Thank you. I think we're almost at the end. Yes. So it's time for Q&A. If you haven't already, if you have a question, please drop it in the chat. I see a couple here. Okay, so I'll read it out. Did you have to modify your ML architecture to handle the increase in data volume as you were scaling, or was it more about optimizing your data infrastructure?
Yeah, it's definitely more about optimizing data infrastructure and increasing the amount of data. So the best performing model actually on a small amount of data carried on being the best performing on a very large amount of data. It just needed like, tons of data to train on, good training systems. Like, we're using, we have like some good data loaders, and I think we're actually flipping to you guys now have like a specific data loader, which we're now flipping to. Before these guys had like a specific data loader, we just kind of written our own ones, but all of this was the key thing in getting our models getting really accurate. We didn't actually adjust the architectures that much. I guess is that we'd only need to really like shift architectures around as we start thinking about like big multi-species alignments and other modalities like integrating attack seek and stuff like this.
I see. Okay. I have a question from Pablo. Hey, Pablo. So, hey, do you have a single TileDB, meaning URI, where you store all your single cell data, or do you have many grouped by certain criteria?
Oh man, the guys on my team are going to tell me off because I'm probably going to get it wrong. I'm pretty sure it's just one. I think we just have a single thingy, like a single mega soma object or whatever. Yeah. I'm pretty sure it's just one big one. Yeah. What we do is we do have a row wise and a column wise variants, I think, which allow us to be a bit faster for certain queries.
Got it. Another one. How do you handle batch effects? I think we covered this a little bit, but how do you handle batch effects when integrating data from different experimental platforms? We've had some challenges when dealing with highly heterogeneous tissue samples.
Yeah, definitely. So this is the one, like, so we figured out that we think this sort of batch adversarially trained single cell variant inference just gets it. If you guys specifically want to help here, just find me on LinkedIn and ping me. We can just show you what we've done. You might just solve it for you guys. The key thing that we found is really one important thing is getting a good, well-balanced training data set, especially one which includes ideally other examples of that technology or that platform that you're looking to integrate data from. So if you've got like just a complete outlier in technology, can be a bit tougher. But if there's other published data sets or anything like even remotely close, that can really help it learn how to sort of align the data properly. Yeah.
All right. Well,
got one more for Pablo.
There's one more. Could you please repeat again, what kind of relational data do you store in a SQL database to complement TileDB? And
we don't know. I was just saying, no, we don't at all. I was just saying we're thinking about using SQL and we didn't. We just chose you guys and we just store it all with TileDB. We have a couple of Excel sheets and that's about it. Yeah.
Great. We really should try and open up the mic. That might be easier, but I'm not sure how to do that. Any other questions? Mayor, am I missing anything?
Outside of that, we have the raffle winners.
All right. Oh. Yes. All right. So actually, before we go to the raffle, I want to just quickly tee up our latest announcement of TileDB Carrara, which is the latest evolution in TileDB platform, and it allows you to, it's the only database designed for discovery. It has all the goodness that Sam was talking about in terms of the data model and the serverless architecture, but then ultimately it supports biotech pharma through the entire data journey of organizing data, structuring it into optimized formats, collaborating with a lot of governance and compliance frameworks. And finally, like analyzing with pre-built workflows, integrated Jupyter Notebook, customizable dashboards. So we're very excited. We will have a webinar about this soon, March 6th, I believe. So please follow that link over there. Well, it's not a QR code over there, and or join the wait list. And yes, now it's time for the raffle. Mayor. All right, we'll reach out to you in short order. Thank you so much, Sam, for sharing all the wonderful details. Good luck on that target. It's so promising. We're so happy. And thanks again, everyone, for joining us to listen in, and we'll see you again in about a month.
Awesome.
Thanks.
Thanks.